hope you can all hear me. I'm Steve Goodman. I'm the Vice Chancellor for Research at the University of Tennessee Health Science Center and a professor there, and President and CEO of the Trials Network of Tennessee. But I'm also the editor-in-chief of the journal Experimental Biology and Medicine, which is the journal for the Society of Experimental Biology and Medicine. And I'd like to start by telling you a little bit about the society and then the journal, and then get right on to introducing our speaker for today. So both the journal and the society were created in 1903. There's a tremendously rich history to the society and journal. The focus of the society is at the intersection of biomedical research and the practice of medicine, and that has been the focus ever since it was created in 1903. And the other focus is the mentoring of the next generation of researchers. I would suggest to you that to at least consider becoming members of the society. The annual stipend to do that, the rates are very reasonable. Regular members, it's $110, postdocs $30, and students are free in terms of membership. And there's a whole lot of things that you get with those very low membership fees. You get an annual subscription to the journal, Experimental Biology and Medicine, which can be electronic or in hard copy or both. You get free page charges for articles published in the journal if you are either the first or the corresponding author, if you're an SEBM member. You can submit abstracts to the experimental biology meetings as an SEBM member. There are travel awards for the student members to come to EB based on review of their submitted abstracts. And probably most importantly, and it's what I've experienced, you're joining a society which immediately provides you with a global networking opportunity. I'm now going to tell you a little bit about the journal. As I mentioned, both the journal and the society were created in 1903. I took over the editor-in-chief role in 2006, and at that time, there were 7 scientific categories. We're now up to 20 scientific categories, and these categories cover really the breadth of experimental biology and medicine. There's virtually nothing that you could be working on as researchers that wouldn't fit into one or more categories. We cover the entire realm of translation from T0 to T4. We have globalized. We now have offices on 5 continents. In addition to myself in the United States, we have global editors in Taiwan, Great Britain, Brazil, and Ghana. And due to the increased numbers of subject areas and the globalization, the number of submitted articles have gone from 200 annual original articles back in 2006 up to about 1,000 or more for this particular year. And so the journal has grown tremendously in this period of time. We've got an international editorial board with 20 associate editors and over 160 members coming from all over the globe. The current 2-year impact factor is greater than 3, and the one that's about to come out will be even higher than that. And so the journal is getting stronger and stronger every year. Concerning this webinar series, we've had an outstanding webinar series. We had one last week. We have another one that is coming up next week on the diagnostics and therapeutics having to do with COVID-19. But today we're going to be hearing from Colleen Johnson. And I know Colleen well because she also comes from the University of Tennessee Health Science Center, where she plays many very important roles. She's Professor and Van Vliet Chair of Excellence in Virology. She's the director of our Regional Biocontainment Laboratory. She's the director of the Institute of Host Pathogen Systems, all at the University of Tennessee Health Science Center. She's extremely well funded, and probably one of them that she's most proud of is an NIH U19 award for $21 million to establish the Center of Excellence for Encephalitic Alphavirus Therapeutics. She studies highly pathogenic viruses, as you'll hear about today, including respiratory, encephalitic, and hemorrhagic fever viruses. Colleen has more than 125 publications and 5 patents, and she's one of the foremost experts on coronavirus and COVID-19 in the USA. And so it's my pleasure to introduce our speaker today, Colleen Johnson. Today's presentation will be COVID-19 antiviral drug discovery. Colleen, you're on.
Thank you very much, Steve. And thank you very much for the invitation today to share with you our research here at UT and in this global effort. So there are so many people working on this now, and we have, of course, joined in this effort. So right now I am going to go to full screen, and hopefully we won't have any issues. So the emergence of new pathogens has been with us as long probably as recorded history. And so this illustration is from a disease called the sweating sickness that impacted human public health in the 1600s. And to this day, we still don't know what the actual causative agent was for this, but if you look at the reviews on sweating sicknesses and what is known, you'll see that it sounds like a lot like coronavirus. So the main area of study that I engage in is in emerging infectious diseases. And when I first started many years ago thinking about emerging infectious diseases, I wanted to understand, well, what does that really mean? And this is a very nice definition. That's a very old definition. And basically, these are diseases that are seen in the population for the very first time, or it's something that's we're detecting for the very first time, but has existed. And in the context of infectious diseases, RNA viruses represent about 70% of all the new emerging viruses that are infectious diseases that we know about now. And so when we look at, say, the last 30 or 40 years, what we see is a number of outbreaks of various RNA viruses over time. These have come and gone. You know, the most recent was Zika, and it seems as if they, most of the time it seems as if they disappear as fast as they appear. However, viruses have the ability to go pandemic. We're seeing it again. We saw it with HIV, hepatitis C, H1N1, and now we're seeing it with SARS-CoV-2. This is what it looks like. So over time, we're seeing an increase in the number of RNA viruses, and we're seeing about a new one every 18 months. So this isn't a problem that once we solve the SARS-CoV-2 will disappear. We are likely to have continued threats as long as we have life on planet Earth. And so this was the map just a couple of years ago of all the various outbreaks on planet Earth. And as you can see, every part of the world is constantly being impacted by RNA viruses. It's continual, the onslaught is heavy. So why? Why are we seeing this? Why does this happen to us? That's a common question that we all ask. In 1918, their solution was, well, you're spitting, so you're creating infectious disease during the 1918. So they put that sign out. That's probably a good sign. So what we know, I mean, there are so many factors. These are just a few of them that result in an emergence of infectious disease. Right now, what we're seeing probably is something that has to do with increased human contact with animals that harbor microbes, right? So in this case, you're looking at consumption of bushmeat and the passage of pathogen from bushmeat to humans. And that's not a unique circumstance. This has been seen with H5N1. This has been seen with many other pathogens. They spill over from nature. And so spillover is this process whereby the pathogen is going about its normal life and it accidentally meets a human, right? And so that's what we mean when we're talking about spillover. But spillover doesn't lead to a pandemic. What leads to a pandemic is the ability of this virus to go from, say, from a bat to maybe another animal, such as a civet, to a human. So that's the spillover event, right? You get spillover, the human is a dead end host, not necessarily dying, but it's not going to be transmitted any further. When you see limited outbreaks that can be from continued chatter of the virus and pathogen in an environment from between animals and humans that result in continual onslaught. However, what we see in limited outbreaks is occasionally a spread from person to person, a limited spread. But when it goes to the second spread, the second human, we don't see continued movement of the virus. It can't adapt. It can't adapt, right? When we see sustained outbreaks, what we're usually seeing is efficient transmission and the beginning of adaptation from human to human. When we reach pandemic, what we've reached is that the virus is in our population. When we reach this point, it means it's probably going to be with us for a very long time. That's the meaning of when it is probably pandemic. So what does that mean? How do these viruses move and adapt? Well, they have to contact new hosts, right? They have to spill over. They have to be able to transmit, and then they have to adapt. And in this little illustration that I drew here many years ago, what we're seeing is that there's going to be a change in the sequence. What we've seen over and over again is that you have the sequence that enables the virus to be fit in its host and have low virulence. But over time, that fitness increases and that virulence, unfortunately, increases in the host. Eventually, when that virus is stable in the population, you may actually go back to this original status. You might actually see that the fitness is high and the virulence is low. That's what the pathogen is aiming for. The pathogen will do better if it has less virulence. And so way back when, right, in 2002 actually was the first case, but in 2003 we saw a sustained transmission for 6 months of the first SARS-CoV. And that originated in Guangdong province. This individual went to a hotel, and from this hotel there were a number of contacts. Very easy to trace. There were only 29 countries, 8,000 persons, but we had a very high mortality. In other words, it was actually pretty easy to trace this virus because of the high mortality. It was much easier than it is right now, right? And so, as you can see, it spread all over the world, but it didn't become a pandemic. It didn't have that sustained transmission, that adaptation in humans. And so this just shows, that situation. So here we are today, or actually this is a couple of days ago. We have we're approaching 5 million cases. We have nearly more than 300,000 deaths globally. How did this happen? The last SARS situation gave us much fewer cases in 6 months. I don't think we're almost at 6 months now, right? And so how did this happen that we, reached this situation? And basically, we can say that this virus was very successful in spillover, adaptation, and transmission to the human host. It doesn't require any animal anymore. So how do these viruses differ? These viruses are remarkably similar. There's very little difference between them. I mean, certainly there's amino acid differences in the proteins, but overall, they are very similar. They both require the ACE2 receptor. They both require cleavage by cellular proteases. So we have a lot of work to do here to understand why this virus is so much more fit and successful in humans. And we have a lot of work to do in that. This is an unsolved problem. And so the point of my speaking with you today is talk about, well, where are we at as a society in addressing antivirals for breaking this chain of transmission and for treating people? Where are we at? And so many years ago, when I became involved in antiviral discovery, I actually was motivated by the lack of antivirals. So when I really started in about 2002, 2003 and engaging in antiviral research in a very meaningful way, I was like, well, where are all the antivirals? Our pipeline is dry. We are trying to make new antivirals for the most part globally. Of course we are now, but basically we have a handful of approved antivirals for just a few viruses: flu, HIV, hepatitis C. For those viruses such as Eastern equine encephalitis, Venezuelan Zika, we don't have any. No drugs are available. And so right now, the entire global community, scientific community, is working on identifying and creating new therapeutics. And the pipeline is wonderfully full of potential therapeutics. And of that, we have, at least of May 6th, which is a couple weeks ago, we had 24 antivirals in the pipeline. So let's take a deep dive and just look at some of those that are being proposed or in clinical trials. Most of them are focused on repurposing. The logic there is if we have something that's already available, we can use it. The caveat with that is that most of these drugs, when you design a small molecule, you design that small molecule for that disease. So more than likely, these drugs, while they may provide some support, are not going to be as effective as a true antiviral, because that's not what they were built for. They were built for something else. And so one of those, though, that we do have that was developed and is now part of the emergency use authorization from the FDA for clinical use is remdesivir. This is a polymerase inhibitor, and it has a very nice in vitro and in vivo activity. And so it's being used now and being evaluated. There is promise, but there is caution, and people are suggesting that this may have benefit. And so all of us would certainly like that to be the case. Right now, I believe the major issue is supplying it for all of the studies that are ongoing in the United States. But there are many other drugs that are being looked at. Probably one of the most spoken about has been chloroquine and hydroxychloroquine, and both of those are in clinical trials. If you look at them in vitro, you can see in vitro with the live SARS virus, you can see that they had very nice EC50s, EC90s. not nanomolar range, but micromolar range, a micromolar, right? Um, so they, out the gate, they don't look bad from an in vitro assay, right? We have Arbidol, which is approved for use in Russia and China. It's been approved for use against influenza since about 1993 in Russia and 2006 in China, and that is being used in clinical trials. There's some other ones that are being used that are anti-protozoal agents. So there's actually a couple of those out there. One I'm going to point out is the nidoxanide, and basically that also micromolar, EC50 micromolar in vitro assays. None of these we would go forward with in a lab focused on antiviral discovery for viruses. We would really want nanomolar EC before we move forward and we get excited. But because of our situation, this is where we are. We're looking at drugs that have micromolar activity and mainly are affecting other pathways, anti-inflammatory pathways, cytokine pathways, endosomal pathways. So we're looking at drugs that are targeting basically host proteins, right? Not the virus itself. In addition to these, we have some other ones that are coming up and have various parts of information available. There's various degrees of stages now in the pipeline for some of these. Baricitinib is being looked at, another endocytosis JAK inhibitor, anti-inflammatory inhibitor, being looked at in trials with and without remdesivir. That's kind of a neat one. Ribavirin and interferon are in trials. We've tested ribavirin. Ribavirin has zero activity in viral E6 cells. Other people have shown that too. So, I mean, it's not one of the drugs we're pursuing here as a control drug. Maybe with interferon, it might have some benefit. It's hard to say. Another one that we've tested here is I always call it T705 because I can't say favipiravir. But that one is also in trials. We've tested it here as suggested in this particular publication that I found it in. It doesn't really have any activity whatsoever against the live virus or in cells, but it is in trials. And it's been primarily used against some other RNA viruses, flu, stuff like that. It's approved. Another one that's in trial is sort of interesting are some of these HIV drugs. And so leponavir and ritonavir are in trials. They don't have very good efficacies, around 15 micromolar. The SI value is very small. So what that is you really want SI values, selective index values that are much broader, right? So that you have more room for dosing. But we'll see what happens with these drugs. And again, there's a couple of various, anti-parasitic, anti-helminthic drugs that are being used with, unknown success at this point, I think. So what we've been focused on at UTHSC and with our collaborative partners, which I'll talk about momentarily, we've been focused on the discovery of new antivirals. So these are good, but eventually we're going to need something better. And so we firmly believe that is possible. We've done it before, we'll do it again, sort of thing. And so what are our challenges? Our challenges are basically that we need to find a druggable target. We need to find something that is a unique target. So by a druggable target, I mean, I'm going to hit that target. I'm not going to hit anything else in the cell. A lot of these compounds I just showed you and some other compounds I will show you have off-target effects. That's going to be complicated. We have to identify properties that make it a good drug. Where do we want this drug to land? That's a big question. Is it just the lung? Is it the kidney? Is it the heart? It's cardiovascular? What are we looking at here? Do we have to make the drug systemic? I mean, we don't know yet. And do we need, then we have children. We have children that have multi-organ response. We're going to have to think about this and how we actually make good drugs. Viruses are complex. So not only do we have to think about how to drug and how to target, we have to figure out when do we target? How can we target? If it's early in the infection process, is that virus as druggable as it is later in the process? Or are we looking at a cytokine storm? Do we need a combination of antiviral and anti-inflammatory? And how do we administer those drugs? If we add anti-inflammatory too soon, we may benefit the virus. That might not be a good thing. That's happened before. We get excited about anti-inflammatory, but it can be dangerous. What we need right now in order to make better drugs is we need to have better data, more pathology, more information on clinical cases. And during these clinical trials that are ongoing, it would be beneficial to understand what the effects are and what is being affected. We also have to realize that we're going to have to understand comorbidities. How are we going to do that? I mean, a patient with diabetes may respond very differently than a patient that has cardiovascular hypertension to the drug that we're interested in using. We also need better testing. That's all we hear about. If we don't have good tests, so if you have a drug, then you have to have a test. You have to have both, and they both have to work perfectly together. Right now, on the news last night in Korea, they said that they're seeing false positives. We have to correct that. We have to correct that soon. So right around March 27th, right after I got over the flu, actually, I had a pretty bad case of flu this year, and it wasn't COVID, but we began our discovery of new or repurposed antivirals. And so I was able to do this quickly because I've been doing it for such a long time. And we've been engaged in lead discovery and preclinical research I've been engaged probably since 2003. And so there's a lot of ways you can approach this. You can go after a biological target, you can do high throughput screening, all this sort of thing, right? And we already had a built-in team. So we already had a built-in team ready to go. And so at first glance, in my early years, I would always go after the target. I'd say, oh, we have to isolate the target. We have to drug the target. We have to make sure. What I found out, we worked on HIV integrase, we worked on Ebola, is that it's hard. It's hard to move the compound that you discover back into a cell. And so what I've done in my later years is we only use live virus. We don't use any engineered virus. We use the virus, whatever the virus is. So we don't tag it. We don't do anything to it. And so the first high throughput screen that I created was back in 2003 and 4, and that was for the SARS coronavirus. And that's this publication here. We used the same approach against flu, West Nile, RSV, and more recently our alpha viruses. This approach that we have moves very quickly. And the reason is because all of these viruses cause cytopathic effect. And so in our model that I created back in 2003 and 4, which is a Vero E6 cell, we can basically target the virus if we use a high enough MOI. And so we can eliminate host off-target type compounds. We basically are going after sort of a pseudo, I guess the best case scenario after biochemical assays. And so cell-based screens have a number of advantages. You already have your cell and the host that you're trying to target, so that's good. You have you already eliminate permeability problems. You eliminate toxicity problems right from the get-go. Metabolism, if you need metabolism, you've got it. And indeed, all of this research is what resulted in the patents that were mentioned earlier. And so our roadmap early on was, how do we do this in level 3? We want to use live virus. How do we do it? And so what we did was we set up plating of all our compounds outside containment, and then we brought our compounds into containment, and then we added the virus. And over time, what we learned was how to look and examine our site. So this is an example of cytopathic effect with RSV, respiratory syncytial virus. And basically what we were looking for is this protection. We're looking for drugs that protect the cells from dying, okay? That's our endpoint. And we've used it successfully. And we have unpublished data on 2 more viruses. We've used it successfully now for 7 times for good solid leads. And so where we're at right now is we've miniaturized my assay from 2007 into 384 well plate. So we could screen 320 compounds at a go in a plate, and we can do 20, 30 plates at this point in time after a month. We used the first isolate of SARS that was isolated. We got that right away in mid-March. We use Vero E6 as our cell line. We use EMEM at 5% FPS, which is very important because you've got to make sure that that works. We had no problem with scale up of cells and virus. For RSV, that was a monumental feat. I think RSV took nearly 2 years to solve how to screen 500,000 compounds. So all the screens I just showed you, including the SARS, we've screened 200, 300 or more thousand compounds during my career. And so the multiplicity of infection we've chosen right now is 0.1, seeding density of 5,000. So we've worked out all these different things. And the length of time to read our assay is 2 days. So we've worked out the conditions to optimize it. And what you want to think about with HTS is this is a puzzle. So once you figure out your MOI, once you figure out your cells, you have to keep retitrating and going back until it all comes together. And the way we evaluate that are with statistical parameters. And this is something that's new for most people. They always want to know, what's your control drug, Colleen? And my control are my negative control is the virus plus cells, and my positive control are the cells. So I'm looking for, that's my 0 and 100. I'm looking for something in between. But these statistical scores tell us for each plate that we run, how good is our plate. So we can say we got to redo the plate or whatever. We also look at signal to background, signal to noise, and we look at our CV values. And that's how we know we have a good assay. And right now we know we're exceeding all these parameters. And so we run a Z plate, and this is basically what I was talking about. This is my virus control. Pretty much everything's dead. And this is my cell control. So what we're measuring here is we're measuring cells indirectly through the amount of ATP. So we use a substrate called cell titer glow. It's pricey, but it works. This is showing the influence of media on cells, and we still have some work to do here. But this is just showing for our West Nile virus assay that a little change in media gives a big change in virus load. So all of these things have to come together. So in other words, if I change my MOI and I change my media, I've got to go back and I've got to figure it out. I can't just change one thing and think it's all going to have the same Z. You know, we have to keep checking. So once you affect one parameter, you're going to affect another parameter. Everything matters. It's like a good, good dinner. And so this is our very first plaque assay that I got in a text message from Jyoti, one of the scientists at our regional biocontainment labs, showing we had the SARS ready to go. So to do this detection on a high throughput manner, I said we use cell titer glow. We do that because we've looked at every endpoint reagent exhaustively over time, over 10 years. I want high signal to noise, high signal to background. Cell titer glow always provides that. There's other ones out there. They may work, they could be unique to each system, but this is the one I've stuck with, and we've just kept with it because of its robustness. You have to have a controlled drug. So I told you you had your positive and negative, but you still want to have some assurance that you're going to see something, right? So here's one of our controls. We have a couple now, and I'll show you a couple little data at the end. This is a commercial compound that I'm not going to talk about, but you can see we have very nice activity at 1 micromolar. So this is a promising lead that actually we tested in animals this past week. So we're constantly been evaluating. And so how do we pick 5,000 cells, right? And so we looked at signal to noise. And so you can see here's a signal to noise with 4,000, 5,000, 6,000. And you say, well, gee, Colleen, why didn't you pick 6,000? Look at that signal. Well, when we look at the Z factor, we started to see more noise with the virus. So we have to balance everything. We have to balance everything. We have to say, okay, let's just keep it at 5,000, and because we're getting more of our statistical parameters there, right? I mean, it's like I said, it's a puzzle. So just because you see a bigger signal doesn't mean your screen is working as well as you think. So here's our strategy. We need flexible media. We need our growth media optimized. We need storage. Everything needs to be easy. We need to have a robust and reproducible path of infection. We've made master stocks of our virus. So Jody, myself, one of my graduate students, Mariah, we've all grown up 400 mils of virus so that we have enough and we have it well characterized. We don't want to be changing virus seed stocks, right? So we're at passage 3. We want as few steps as possible. No washing, no rinsing. We just basically, at 2 days, we had cell titer glow. Half an hour later, we read, we're done. And so this is where we're at. You know, we're adding our compounds. So we've actually changed up what we have done over the years. We're adding the compound the day before. Then we're adding cells. We plate for 2 hours in the level 3 lab, and then we add virus. And then 2 days later, we read. And so this is what our plate setup looks like. It's a typical format. We get 320 compounds in there, and we can run a variety of formats. We titrated our DMSO concentration because all drugs come with DMSO. So this is when you bring it all together, then you're ready to start screening, right? So you got to do all that work, right? Which was really remarkable effort in my lab and the RBL staff working together. And so you see this for West now. We run a series of plates. And so you have your cells, your positive control, you have your virus control, and then you have your drug, right? And so this suggests that that's kind of telling you what you might see with a good drug. And I think this was ribavirin, so it wasn't really good at that point. So here's what it looked like way back when. This is my SARS-CoV-1 example. We had screened, once we did our Z data analysis of our plates, and we have very similar data right now. We have very similar signal to noise, signal to background, and Z. Our Zs are, and I'm going to show you in a minute. We screened more than 300,000 compounds back then. Did we miss anything? I don't know. We may have. But none of those advanced. So unfortunately, the interest in SARS waned, right? And we moved on to alpha viruses and other viruses. So we actually have these hits, and we are looking at some of them now. But this is what we've done in the last month. We've screened well over this says 41,000. We screened probably closer to 50,000 compounds in the last 4 weeks. We ramped up slowly to make sure everything was working. And so what you see here in this type of analysis that was done by another group, you see your negative control that I mentioned to you, your positive control, your live cells. All the black dots are all the things that didn't work, right? And then you have all the beautiful little blue dots, right? And so in the next 2 weeks, we're going to be screening those blue dots, right? We've screened some blue dots, but we don't have all blue dots here to share. As we've been moving forward, we're assessing our Z factor. That's extremely important. We want to make sure that our Z factor, I prefer it above 0.5, but you can see we've had a few drop below. But again, this is a process of growing numerous T150 flasks on a constant basis, getting the cells just right, replating the cells. We plate the cells robotically. We plate the compounds. Some are plated robotically, some are plated by hand. So it just depends what we're doing. But the Z prime factors are looking very, very good. So what are we going to do? So this is our roadmap. This is one of the ones we had for H5N1 many years ago. And so basically, we're going to continue to screen libraries with a lot of collaborators. We have actually 2 more collaborators this week that have libraries that they're bringing in. Once we find our hits, we're going to be screening them, and we've screened a number, and I'll share that with you in a second. And then we're going to be looking at other types of studies like time of addition. Does this compound affect before or after screening? So what we've been spending quite a bit of time on is working with Oak Ridge and UT and University of Alabama, Huntsville, along with my long-term collaborator, Jennifer Golden, in can we find compounds that have been repurposed? So what I just told you was we have been screening a lot of compounds. Doesn't matter what they are, we screen them, look for hits. But this is a more methodical. So Jeremy and Jerome are taking virus structures. They're creating molecular simulations. They're docking compounds virtually and then providing us compounds that could be potentially of interest. Jennifer is reviewing the compounds that Jeremy identifies and Jerome. And then we remove some of those based on they probably wouldn't go forward anyway. And then we screen them using a dose response, because now we're filtering, we're taking 100, 200. So that's, not even a day's work, right? And so here's one of them that we've identified. It's has a beautiful EC50 graph. It's called Cinecolit. It's an Amgen drug, so we'll have to do next steps. So we have a nice dose response from this, as you can see. I mean, it's as beautiful can get. And we're going to have to take this compound now, this FDA-approved compound, and look at titer reduction, look at order of addition. And then if those look good, we'll take it into our hamster model, which I'll share with you in just a moment. Another one that we've identified is actually has a brand name, Nexavar, our sorafenib, and this is a multi-kinase inhibitor. And while it looks good, it had micromolar activity, as everyone is seeing with these FDA-approved drugs. Look at all the targets it hits. It hits like everything. This is going to be a hard one. This is going to be a hard one to move forward. So this is what I meant. You can screen drugs, but if they've got a lot of effect everywhere else, it may not work out for you. However, because this is FDA approved, it could work. So, but we won't know until we get to the animal. So we can run in vitro assays all day long, right? But the proof comes when you go to the animal. That's when the rubber hits the road, as they say. And so in addition to what we've been doing in antiviral drug discovery, we have worked very hard on animal models. Originally, we thought we could maybe had some success adapting the virus to a mouse model with a knockout and different knockouts. We use this knockout, that knockout, and that, you know. And finally, we just said, no, that's not going to work. And a group in Hong Kong published an animal model with the hamster. And so this is our first proof of concept that the hamster model in our hands works as well as it did in the group at Hong Kong. And so we've run PBS and we've run 2 doses of the SARS coronavirus, both 10 to the 3rd and 10 to the 5th. And we've looked extensively for on day 2 and 4 post-infection in lung and trachea to look at virus load. And then on day 7, we looked at the histopathology, right? And so I don't have the histopathology with you ready for prime time, although it is pretty much ready for prime time, but we need to do a little bit more there. What I can show you is that the model repeated beautifully in our hands. We saw a very similar decrease at the higher dose with in the hamster model. So these hamsters were dosed intranasally and they lost weight over time. We took it out to day 7 because according to that publication, that's where they saw the highest amount of pathology. I do know from others that the hamster has a very hard time recovering even out to day 12, 13, 14. And so we're now looking at taking this out longer as well and looking at the pathology over time, because those will be very important endpoints for understanding whether or not our drug works. So right now we can use weight, and we can also use, let me just see here. We can also use virus titer. And so this is showing you virus titer. And so lung in day 2 and day 4 is in blue and green. And so if you look at the low dose and the high dose, what you see is you see a relatively similar amount of virus in the lung. And so I would assume days 2, 3, and 4 would be somewhat similar, although day 3 may have a higher virus load. We looked in trachea. That's going to be more variable depending on where it was cut during when we were taking it. But you can see that there was plenty of virus in the trachea. I'm a bit surprised we didn't see much in the nasal turbinate. I assumed we would see because of the olfactory issues that have been noted with people having lost a sense of smell. But we're very pleased with the model, and as I mentioned, we have started testing drugs in this model, and we'll be starting testing a number of drugs over the ensuing weeks. We have a number of drugs lined up for testing in the hamster. So it's just going to be hamster, and then for until about July, and in July we'll get the ACE2 human ACE2 mice from Jack's labs. It's reported from Adolfo Garcia on Tuesday that infection of the Jack's mice resulted in lethality with the same strain that we have. So that'll be a lethal model. The virus did invade the brain and the lung. In our model, there was no apparent pathology in the brain. We haven't looked for virus yet. We would assume there isn't any, especially since the olfactory nerve didn't show anything. So basically, my conclusions or my advice or whatever you want to call it is you have to begin with the end in mind when you're thinking about drug discovery. It's really important to have the roadmap and to have a good, well, I'll jump to my 4th conclusion. It's important to have a good medicinal chemist with you step by step. To look at your compounds that you're identifying early before you get too invested. And so I want to thank all of the crew at our regional biocontainment lab that have been very dedicated to this effort. It's just been remarkable how they've really stepped up to get this done during this time. And this is our crew that have been leading all the different studies. Dong has been leading a lot of the hamster studies with Lillian and Jody and Jasper and Shruti have been leading a lot of the small molecule screening. They've done a fantastic job. Yi's been running all of our pathology that I didn't get a chance to show you. And so it's really been quite an effort, but not here shown is Walter, who's been plating all the compounds, Courtney, who's been growing all of our cells, Ying Kong, who's joined the team to look at the mouse, and then Elizabeth Patrick, who's looking at the immune response and interpretation of the immune cells we're seeing. And so I want to thank the University of Tennessee Health Science Center for the funding that they provided for us from the Office of the Chancellor. It's been really remarkable to have this support so that we can contribute to this effort. And so I believe that's the end. And so this cartoon I had for a very long time. And I don't think anybody really has ever understood it, but maybe people do now. Basically, it says, Captain has turned off the infectious disease light. You're now free to move about the cabin. That's probably coming after they decontaminate us. Anyway, thanks, and I'm happy to take questions.
So thank you, Colleen. That was absolutely brilliant. And I think we're now going to open it up to questions.
Yes, so good afternoon, everyone. My name is Natalie Fuentes. I'm a postdoctoral fellow at NIH and also a member of the SCVM Training Council. So I will be leading the Q&A answer session. Dr. Johnson, thank you very much for such a great presentation. It was amazing, very clear, straightforward. I think a lot of our audience actually got a lot of information out of it. So we have one question from Facebook. Ben Zimmer wants to ask you, how well do in vitro compounds translate to in vivo activity?
It's a great question. It's very difficult to predict. It's very difficult to predict. When we get into nanomolar activity, I think we have better confidence. But if our drug doesn't target the site where the virus is, then it's going to be difficult. So with those two pieces of information, we can have better confidence. And so usually I work very closely with someone who does the pharmacokinetics. Pharmacokinetics provides us a lot of good information about how much of the compound got into the animal, where is the compound going. We might do a biodistribution study. And with those two in hand, then we can plan our in vivo studies better. But yeah, it's very difficult. There's no crystal ball or easy way path.
Great. Another question. So an important part of drug discovery are clinical trials. How are clinical trials being impacted by the COVID-19 pandemic? For example, challenges such as self-isolation, site closures, travel limitations, in-person meetings may arise. So how is this actually affecting research?
Yeah, so I think that research certainly has been impacted, but from the phone calls that I've been engaged in, I think that there are many labs that are open and conducting research either at preclinical or phase 1, phase 2, phase 3. There's a lot of companies that are engaged in phase 1, 2, 3 trials, and so they're organizing those globally. So there's actually an enormous amount of clinical trials that are ongoing. I think there are hundreds of them now. I went to NIH's website to update my slides on a few of these that I was interested in. And it was just every country is trying some of their, the ones they're expecting to move forward based on their internal research, So I think, from my opinion, people are making amazing progress. When the 2002 outbreak of SARS occurred, we were amazed that we had the virus in a month. I think we're all going to be amazed that we already started vaccine trials, in 5 months? I think we're all going to this is a new milestone for the general scientific community that we could move this quickly. And the Moderna vaccine looks very promising with the spike. I mean, a lot of people showed boost to the spike protein that the mRNA makes. So it's all looking very good.
Great. Now we have another question from Dr. Stanbrook. How frequently does the virus incur mutations, for example, in the sequence encoding the spike program? And do mutations render the virus either more or less virulent?
Right. So in our roundtable discussion on Tuesday with the NIH, we had a presentation from a lab in Singapore on that very question. And for the most part, there is still only one species or strain of this virus. No one has any data yet that any of the so-called mutants are having an impact on fitness. So I think those stories are coming. I'm sure people are working on them. But there isn't, the general consensus from that conversation was no one had any evidence that any of the mutations are. And so what you want to think about when you're hearing all this is, when did they get that virus isolate? Where is that virus isolate from? Because you're going to always see some drift and some different genotypes, basically genotypes. And it's going to be rare that they're going to change even further. We hope that doesn't happen, that there's no more lethality associated with this virus. That it's going to go towards, remember I told you in that first slide, I'm hoping it goes towards less fitness. I mean, not less fitness, more fitness with less virulence, right? So that's what I would suggest. They usually eventually move along that trajectory.
Okay, thank you. So we have another question from Facebook, from Grecia Hernandez. In your model, you have up to 7 days post-infection, where you see most of the viral symptomatic effects in terms of timeline. How that translates to a real patient exposure to the virus?
And it's a great question. We've only taken out to day 7, but I do know from other people's studies that the pathology that we saw, which was quite serious on day 7, continues out to day 10. So it looks like from day 6 to day 10, the lung is seriously compromised. We see emphysema like morphology in the lung, perhaps. We're not, I mean, a lot of this is very new, right? So we're trying to interpret our findings. We're seeing coagulation in the lung. We're seeing a lot of immune cell infiltrates. The lungs do not look, from our perspective on day 7, the oxygen capacity of the hamster is seriously compromised. But we're going to continue on doing serial sacrifices to understand more.
Great. Also from Gracia, at what time point during virus infection inside the host or patient, when do you think these drugs may be more suitable to treat patients?
Early. If it's an antiviral, early is going to be good. That's going to be good. I'm really worried about anti-inflammatories being used early, but I think the sooner you can get an antiviral, an actual true antiviral, that means a compound that affects, when I'm saying this, I'm saying a compound that targets the virus. If you can get that sooner than later, I think that's going to be your best bet. Most of these drugs that have been used so far are decreasing the duration of symptoms by 2 days, 1 day, but not significantly.
Thank you. Now we have a question from Samuel from University of Ghana. Since there are new drugs in clinical trials, are you planning of using more of these drugs as your control in your work?
Yes, we have remdesivir, and we'll be looking at it in our next hamster study that starts next week. So
Great.
hopefully we see everything's beautiful.
So Dr. Stanbrook also is asking, does the viral RNA undergo epigenetic modification? Can enhancement of epigenetic modification be used as a therapeutic approach to reduce virulence?
Well, certainly if you want to make a traditional vaccine, you could passage the virus many times to reduce virulence. That's a sort of a common old approach to making a good virus vaccine, just to passage it over time in cells.
So I think I will, 2 more questions and we're gonna let you go because it's already an hour. However, you mentioned hamsters, the use of hamsters as a model. Maybe this is a little bit early in time in terms of what your research is about, but have you guys considered using a specific sex of the animal, you know, like female, male, or a specific age?
Yeah, we've used both. We've used males and females. We don't see any difference. And the age of these hamsters are about 6 weeks old. Great question. I should have said that. The hamster, unfortunately, we have to get them in when they're 3 weeks of age, or we were trying to, because they're fighters, and then we're trying to adapt them to our environment. So that's been a bit challenging. Yeah, they're young.
And our last question, so you mentioned for initial screening that you use the Viro E6 cells, which are kidney epithelial cells from, I think, African monkey. I understand that these cells are interferon deficient, but they still have interferon receptors. So why were these cells selected per se, and will it be important to test other, like the candidates with other type of cells, for example, lung cells or even just human cells for that initial screening?
Yeah, so there are reagents available. I've other cell lines that are being made available. One is A549 with the ACE2 receptor, and so that's a human cell line. I like the Viro because it doesn't have much of a response. I'm really looking for those candidates that target the virus and not the host. So it's really worked for me well. It has a very strong cytopathic effect for this virus. Other cell lines like the Viro CCL81 or whatever it is doesn't. In other cell lines, you'll see you will not see cytopathic effect. You'll see syncytia, and that won't work for the screening. We need a really strong cytopathic effect. Hit them hard with the virus so that you can find drugs to the virus. So that works really well. I was trying to say earlier, it's kind of like these Viro E6 cells are like my biochemical assay, right? I mean, to me, it's a minimalistic type of environment. It's as minimalistic as I can go. We've used primary cells. Primary cells, of course, are very difficult. Calou cells we've used. They don't, they grow real slow. Yeah, so for me, I love these cells. These cells for the screening part.
All right, thank you very much.
You're welcome. Thank you for all the great questions. Great questions.
Thank you, Colleen. This has been terrific. And I want to thank the audience as well.
Let's find a drug.
Thank you all.
Thank you.
Thank you all so much, and we hope you join us for next week's third edition of COVID-19 webinars. We'll follow up after this with a survey, and if you need to refer back to this webinar today, it will be online at scbm.org. Thank you guys so much for everything, and we'll see you next week, hopefully.
Thanks for the invitation.
Of course, thank you.
Bye.